Welcome to Milica's proposal defense. Milica, I think you're going to see that Milica has a really cool project, a very, very challenging project. And as her committee chair, the first thing I'm going to do is thank the committee members who are taking up my slack in many ways by helping her and teaching her new things. Just to realize how challenging this was for her, she's now immersed in the world of molecular approaches and biogeochemistry and trace gas loss and leaching and hydrology and everything else. But putting it best, it was Steve Running, who's at University of Montana. And Steve, I knew him as a graduate student, and I'll point out that Steve won a Nobel Prize. He was part of the IPCC group who won the Nobel Prize. And Milica, in his words, comes to us from the world of frog squeezing. And so no offense to any frog squeezers out there, Erica and whatever, but it's a big change going from the type of work she was doing. Thanks, Erica. From the type of the work she's doing to now really taking a whole agroecosystem approach. So we will have the seminar, and then because of scheduling, there'll be a break. So questions can run a little long, but the committee then will reconvene an hour later. And I sent out a link for that because it's the rules that she not set that up. So with that, then go ahead, Milica.
Okay, awesome. Thanks for the introduction, Dave, and thank you to everyone that has joined today. I'm really excited to share this with you. It's going to include some of my research already, but then also plans of what I expect to do. So I'm going to be looking at soil microbial transformations and substrate limitations as drivers of reactive nitrogen loss from agroecosystems. So for a quick outline, first I'm going to talk about what reactive nitrogen is, why it's important and why we should care. Talk about what the objectives of my dissertation are, what the hypotheses approach, what the expected results are, and then also what the significance is. Then I'll talk about the experimental designs that we have set up, as well as some initial findings. So nitrogen is an important component of all organic matter and in many biological processes, such as photosynthesis. And about 80% of our atmosphere is composed of nitrogen in the form of dinitrogen, or N2. But this form is biologically unavailable. And there's only a few natural pathways that can input a large amount of reactive nitrogen into soils for them to be productive. One of these methods is dry and wet atmospheric deposition from already available nitrogen in the atmosphere. Another is biological nitrogen fixation. For a long time, biological nitrogen fixation was used to increase nitrogen availability in agricultural soils, which then enhanced photosynthetic rates, increased crop growth, and yields. But this method did limit how much reactive nitrogen could be added to soils, and was a large factor in preventing the global population growth that we have observed in the last 100 years. But in the early 1900s, two German scientists invented the Haber-Bosch process, which had the ability to industrially capture this unavailable nitrogen from the atmosphere, transform it into the fertilizer that we use today so we can enhance crop growth. And this obviously led to a strong relationship between fertilizer, food production, and now there's a drastic increase, and there's been a drastic increase on global dependence on fertilizer. So if you look here on the x-axis, we have time. This first triangle is when the Haber-Bosch process was invented. The second is when it became internationally distributed. On the right side, we have the right axis refers to lines that are associated with industrially derived fertilizer. The left side is global population. So if we follow this purple line, you could see following the international distribution of fertilizer, of this method, you can see that industrially derived fertilizer skyrocketed. But then you could also see that the amount of fertilizer that we were using that was actually coming from this process also increased to where it's almost at 100% always now. Then you could look at global population before fertilizer use. You could see that population was kind of growing at a pretty slow, steady rate here in the red. And then you could see that following international use of the process, this population growth increased. Another important thing to mention is that here, this blue line is the number of people on the globe that are actually supported by fertilizer use. And in the 2000s, it was about 50%, and it still continues to be 50% today. And this sort of relationship has created a very strong feedback loop, positive feedback loop, where when there's increases in food availability, that allows the population to increase, but that also causes more food demand, which requires us to grow more crops. And to meet that new food demand, we have started inputting more and more industrially fixed nitrogen fertilizer in order to meet crop demands. But increases in reactive soil NitroGen also lead to loss through processes such as leaching of nitrate. This is, or NO3, and this is particularly important because this nitrogen compound can cause eutrophication, can contaminate groundwater, which is particularly important here, can cause human health decline, and can lead to economic loss. Nitrogen is also lost as a number of gases, including ammonia, nitric oxide, nitrous oxide, and dinitrogen, that unreactive form that everything started with. And particularly important in nitrous oxide because it's a highly reactive gas in both parts of in the troposphere and stratosphere. It is also a very potent greenhouse gas, and it has the ability to really affect human health. Ammonia is also emitted from agricultural soils, but slightly acidic conditions tend to lower that, and that's what we're seeing in our region. And nitric oxide is also a very reactive gas that can be released at high magnitudes, but that would be a dissertation on its own if looking at that gas. And so we know that in order for any activity to commence, we need a substrate and favorable conditions. So when fertilizer is applied, it creates this pool of ammonium, or NH4. This pool can then undergo its first biological processes called nitrification. This process takes ammonium and converts it to nitrate, or NO3, that nitrogen compound I said leaches out of the system. During this, an intermediate in nitrification has the ability to break down and create nitrous oxide. Then a new nitrate pool is formed. This new nitrate pool can also be used by plants or crops, or can be leached out of the system, or can undergo more denitrification steps. And for the next denitrification steps, I have classified them into two groups. The first group is incomplete denitrification. And here, this nitrate only gets reduced down to nitrous oxide. It doesn't go any further. Then, and during this process, nitrous oxide is an intermediate in denitrification because it will get formed if the process goes to completion. The second form of denitrification I've identified is complete denitrification, and that is the final reduction of nitrous oxide to its inert gas, dinitrogen. And so we know what the main biological processes are, and we know what their abiotic products, and we have been able to trace the abiotic products pretty well. But the problem is that we don't really know which process is active and producing nitrous oxide in real time. So therefore, my objective is to address how substrate limitation and soil microbial composition, how they control nitrous oxide and dinitrogen flux. It is difficult to identify which microbial groups are active because they both release nitrous oxide as a product. So if we're just tracing that gas, both processes could be producing it. But the process that's dominant could also change instantaneously through the induction of a hot moment. Hot moments are short bursts of elevated activity that are easily distinguished from lower and longer background activity. Hot moments are induced by changes in the soil environment that are specific to each community. So for example, nitrification requires an ammonium pool and requires soil moisture for there to be moisture, but still a little bit of aeration. And same for denitrification. They need the nitrate substrate or the nitrous oxide substrate, and they need more anaerobic conditions as the process goes on, so higher soil moisture. Okay, so based on this, my first objective is to address how substrate limitation affects microbial communities responsible for soil nitrogen transformations. And as I mentioned, there's 3 communities of interest. The first is soil nitrifiers, which will be presented in purple for the rest of this presentation. We have incomplete denitrifiers, which will be in blue, and we'll have complete denitrifiers, which will be in red. And I hypothesize that soil nitrifiers will be dominant in early fall and summer. And this is because the substrates required by this microbial community are present at higher concentrations during those times, and soil environmental conditions are more favorable. Next, I hypothesize that incomplete denitrifiers will be dominant in the late fall and spring, following a spike of activity from nitrifiers, because as I've mentioned, nitrifiers create a substrate for denitrifiers. And finally, I hypothesize that complete denitrifiers will be dominant in late fall and early spring, following incomplete denitrification and further induction of very anaerobic conditions. And overall, it is very difficult to identify individual species responsible for nitrogen transformations because many organisms actually have the ability to transform soil nitrogen. And this includes organisms from each of the major domains. So therefore, I am proposing to classify soil microbial communities based on their functions. And these functions will be identified by functional gene groups. So for example, active nitrifier groups will be detected by a functional gene group responsible for coding for ammonium monooxygenase. This group is specific to nitrifiers. So if we see in the activity of this gene increase, then we can start suggesting that nitrifiers are the lead role. For incomplete denitrification, it's nitrate reductase. And for complete denitrification, it's nitrous oxide reductase. And just for all of them, these groups are only classified by these functional genes. And I will be extracting RNA from soils to look at which communities are actually active in lab and field settings. So why is this significant? First of all, this is significant because traditional identification methods used functional gene groups that have over or underestimated contribution from the different identifiers. And this is because the functional gene group that has historically been used to identify denitrification has been nitrite reductase, right here. But recent work has found that soil nitrifiers, under certain environmental conditions, can produce nitrite reductase at very, very high levels. This process can also start being the main form of nitrous oxide being produced. So every single time we were thinking it was denitrification could have also been nitrification or nitrifiers, which would require a very different management technique. Next, typically the active community members are not identified in soil systems. Typically DNA is analyzed, and when you're looking at DNA, you're looking at who is all there. This can particularly be a problem with the nitrogen cycle because of the hot moments that I mentioned. So there's going to be this larger community there, but their activity won't start increasing until they reach favorable environmental conditions. And finally, successful management practices cannot be suggested or implemented if we cannot identify the biological sources of the gas and then determine how environmental conditions are affecting those groups, because that is what we can hopefully control. Okay, so I know there's a lot going on here, and I expect there to be great variation, but I'm only going to concentrate on 4 key points of the year where substrate and soil moisture play a large role. So here in the first part, actually, so on the x-axis, we have day of the year. We have the months labeled. Here, this green line is approximate time of fertilizer application. The red dashes is N2O flux. Solid black line is nitrifiers. Dotted black line is incomplete denitrifiers. And dashed black line is complete, with the relative magnitude of each of these variables on the y-axis. Okay, so at the first time point, I expect nitrifiers to have the lowest activity because they are less temperature tolerant compared to the both groups of denitrifiers. I expect there to be a steady increase in N2O flux as temperatures are increasing, and I expect the complete denitrifiers to increase in prevalence and activity when increasingly waterlogged conditions are induced, which can happen as winter is starting or as it is finishing. So then in the springtime, we have multiple thawing events, but instead of putting an arrow for all of them, I'm kind of just going to use this one and say there's multiple thawing events. So during the main snow thawing events, complete denitrifiers will be the main active community. N2O flux will start decreasing because it is being consumed to create N2 by complete denitrifiers. Additionally, nitrifiers will also start increasing in activity because their substrate will start being naturally produced during this time through processes like mineralization. Then the late summer months will cause all activity to start slowing down due to substrate limitations and dry conditions. Overall, I predict nitrifiers will still have higher activity because they can survive in these lower soil moisture environments, and ammonium is more likely to be present in the soil. And finally, following fall fertilization, which adds a substrate for nitrifiers, nitrifier activity will increase greatly. And then once they produce substrate for incomplete denitrifiers and there's precipitation increases, their activity is going to increase as well. And then complete denitrifiers will have to lag behind incompletes as they are the ones that create the substrate for the next step. So overall, I predict that the active members of the community will directly correlate with nitrogen transformations that are occurring and also will be influenced by environmental conditions and substrate availability. Okay, so my second objective is to determine how substrate limitation affects the biological mechanisms responsible for N2O and N2 emissions. The biological sources of the gas will be determined by substrate availability and environmental conditions. So if we look at this figure right here, you could see that on the x-axis, we have water-filled pore space. And you can think of water-filled pore space as a proxy for soil moisture. So higher percentages suggest that the soil is more wet. On the y-axis, we have the net flux of nitrogen gases, which are shown here. We're not focusing on nitric oxide, but it gets produced as well. And so you could see that nitrifiers, they have a range of moisture levels that they like, but they'll only start producing N2O if they'll only start working if ammonium is present. Denitrifiers, incomplete denitrifiers, the same thing. They'll only start working if those soil moisture conditions are induced or if nitrate becomes available. And for complete denitrifiers, the same thing, except that they require N2O as a substrate and require very, very anaerobic conditions. So I hypothesize that nitrification will be the main source of nitrous oxide in the fall. And this is because ammonium substrate becomes available following fertilization, and the soils generally have a water-filled pore space range that fits nitrifiers. Denitrification will be the main source of N2O in the fall and spring. And this is incomplete. And then finally, N2 will be produced in early spring, and it will originate from complete denitrification based on environmental conditions that are expected during this time. And ultimately, I think that the majority of nitrogen will be lost from denitrification if we look at it from an annual basis. And this is for multiple reasons. One of them is studies that have induced favorable substrate conditions and microenvironment conditions for nitrifiers, and then the same thing for denitrifiers. And when they have allowed them to produce N2O, denitrifiers typically produce more. In addition, denitrification has nitrous oxide as an intermediate. So this means if complete denitrification is occurring, most likely nitrous oxide is produced. This is different from it being a byproduct where the intermediate in nitrification has to break down to create nitrous oxide. And finally, the soil, the bulk soil may seem less moist as time goes on, but there are small parts of soil called microsites that can have highly anaerobic conditions, basically waterlogged, and they have found that there is higher rates of denitrification in those microsites. Okay, so N2O will be collected in field and lab settings and will be analyzed for site-specific nitrogen stable isotope composition of this really cool linear gas. All of these are pretty scary, but I'll talk about this in detail in a little bit, and hopefully then it'll become a little more clear. But the two different sites in this gas are the alpha, or the central nitrogen, or the beta, or the terminal nitrogen. In addition, we will measure dinitrogen and N2O emission that is derived from fertilizer. But we will need the help of isotopically enriched substrates. And we need this because we will not be able to detect N2 emission regardless of magnitude if we aren't using a tracer, because of the 80% in the atmosphere, it drowns it all out. And then I will monitor background and hot moment activity to determine which microbial processes are actually creating the gases during this time. Okay, so before we delve any further, I want to provide a brief primer on traditional bulk stable isotope analysis and this novel site-specific stable isotope analysis. So traditional stable isotope analysis takes advantage of naturally occurring variations in abundances of isotopes of many different elements. And these isotopes can be used because they do not break down, or they have not been seen to break down through all of Earth's history. And they can be used as a tracer for biotic and abiotic processes. If you look here, in existence, the majority of the nitrogens are lighter. Most of them are 14N. And a very small amount is the heavier isotope. And this is typically seen with all the different elements. And when you're tracking variation due to different processes, you look at the ratio of the heavy to light isotope in the sample. And here, that is represented by a 15R sample. This number is obviously going to be very small if you look at these percentages. So to make it easier to work with, no, this number is going to be very small, and it will change between labs as well as mass specs or RUMs, depending on multiple factors. So to make everything uniform and easier to work with, the values are overall reported in the delta formation in units of per mil. And then to put everything on an international scale, there are certain products whose values have been determined and accepted internationally, and their ratio of heavy to light is here on the bottom. Okay, so then when you see higher values of delta 15N, that means it has more of the heavy isotope. When you see lower values, it's more of the lighter isotope. So the variation in heavy to light atoms in biomass occurs due to a process called fractionation. And this process, preferentially uses, due to this process, lighter isotopes are preferentially used and taken up into the products. So this usually leads to products having lower delta 15N values compared to the substrates. And the shift is called discrimination, and values or ranges of values have been identified for different microbial transformations, including nitrification and denitrification. And this technique is still being used today and is still very popular. The only problem with it is that this approach requires us to have the delta 15N value of the substrate. So we need to have the delta 15N value of the nitrate or the nitric oxide. Obviously, there's a few problems with that. There could be space constraints, time constraints. As I said, nitrous oxide leaches readily, so what if it leaches out of the soil before we're able to analyze it? However, and this is pretty intense slide, but I'll go slowly through it. These constraints can be overcome using a novel approach called site preference. So this approach takes advantage of the fact that enzymes responsible for creating nitrous oxide from nitrification or denitrification will always cause the same amount of fractionation at either the alpha or beta position, regardless of substrate composition. So for nitrification, N2O is produced by an enzyme called hydroxylamine oxidoreductase. And this reduces this intermediate in nitrification hydroxylamine and produces nitrification. This enzyme, if we move over here, this enzyme has one binding site, and recent work has found that the beta nitrogen typically binds to the binding site first. Followed by that, the alpha nitrogen attaches to the beta. And as I already said, there are already more 14N atoms overall than 15N. So the chance of the first atom attacking being 14N is great. But then also when it comes to remove the final oxygen to create nitrous oxide, the atoms having the lowest energy bond tend to break. So in this case, that would be the bond between the 14N and the oxygen. And this in the long run results the beta values being smaller than the alpha values. During denitrification, a different enzyme is used. The enzyme is nitric oxide reductase, and that transforms two nitric oxide molecules into one nitrous oxide molecule. Nitric oxide reductase is a little bit different. It has two binding sites. And so this is pretty new work still. Scientists we're all still trying to wrap our minds around this. But the most recent work has suggested that when nitric oxides bind with nitric oxide reductase, two nitric oxide molecules bind at the same time. They're released from the cofactor at the same time, and they lose their oxygen at the same time. And with that, that results in the values at the alpha and beta position being very similar to each other. Okay, so then if you look at site preference is the difference between the two nitrogen atoms, the two nitrogen positions, so the alpha minus the beta. And this difference will stay the same regardless of what the substrate is. This is similar to the C3 and C4 photosynthetic pathways for plants. Different enzymes are used and the values will stay pretty much the same, similar. End member values for site preference were determined using pure bacterial cultures. So for example, when we were looking for denitrification values, pure cultures or denitrifiers were looked at, and values between -10 to 0 per mil were consistently observed. For nitrification, same thing. Pure nitrifiers were raised, and values range from 33 to 40 per mil. But in natural settings, we don't just have nitrifiers or denitrifiers active. We actually have a consortium of bacteria. So we're going to have multiple species that have the ability to do the same function in the system. So our samples will be a mix of these two values, and we'll have to partition out how much each contributes. Okay, so based on this, and based on the argument that substrate availability and soil moisture are very large drivers of the processes, I expect increases in soil moisture or water-filled pore space to increase the activity of incomplete denitrifiers, and then later with increases in soil moisture, increased activity of complete denitrifiers. I expect site preference values to keep decreasing to suggest reach values closer to -10 to 0, suggesting denitrification is more prevalent. In a closed system where we would run out of substrate, we would see the site preference values start increasing if the final reduction step is occurring. And that again is because the substrate stays heavier, so we're going to have an increase in site preference, and the N2 will be lighter. And finally, an increase in water-filled pore space. leads to a decrease in the N2O over N2O plus N2 ratio. This ratio is used as a proxy to measure N2 loss. And so this ratio will become smaller when complete denitrification is active because more N2 is being produced and N2O is being consumed. So that will change values in the numerator and denominator. But on the other hand, with lower soil moisture, I expect to see increasing activity in soil nitrifiers. Obviously, if it's too dry, they won't be active as well. I expect to see increasing site preference values, and I expect to see an increase in the ratio. Okay, so it is important to characterize N2 emission to its biological origin because any loss of nitrogen results in economic loss because the crops aren't able to use the nutrients that are available through the fertilizer addition. And N2O gas is particularly important because it has very strong impacts on human environmental health. In the troposphere, N2O plays a large role in creating smog. So in LA, the smog that you see, some of it is due to tropospheric contractions of N2O, and that can lead to cardiovascular problems. In the stratosphere, on the other hand, so that's higher up in the atmosphere, N2O is an extremely potent greenhouse gas. It is 300 times stronger than carbon dioxide. So if we have 300 carbon dioxide molecules, they have the same strength as one N2O molecule. In addition, N2O is highly reactive in troposphere and is a leading cause of stratospheric ozone destruction. So the ozone that's protecting the globe. And then this gas, since it's very reactive, it will undergo numerous transformations that can increase acid rain events. And finally, we were missing a fundamental understanding about which biological mechanisms are driving this nitrogen loss. And without this understanding, we cannot work to minimize that loss. So the biological mechanisms driving these losses, they require different settings. And if we don't know which ones are active when, again, we can't prevent the establishment of those settings. Also, models many times can be used to predict when to fertilize to prevent nitrogen loss, and will be especially important when predicting future climatic scenarios. And without understanding of where nitrogen loss is, these models won't be as robust. Okay, finally to the experimental design. So all of the field experiments will be conducted at the Palouse Conservation Field Station, or PCFS. Out there right now, we have an automatic chamber system. It measures N2O and CO2 gas flux from small microplots that we established in the fall of 2018. They measure the gas emission from there at every 3 hours. So we're getting really high temporal resolution. This is what the site setup looks like, and then this is what the individual chambers look like. So in addition to us getting concentration, we're also getting the site-specific stable isotope composition that I mentioned of the gas that's being collected. We're also getting soil moisture and temperature for the 5 top centimeters, measured at 5-minute intervals, and atmospheric conditions are recorded from AgWeatherNet. In addition to these field experiments, I will also run control labs experiments using intact soil cores from the field. And for this, we won't be able to have continuous measurements, but time points of interest have been identified to calculate flux there and identify the biological origin. Okay, so the first experiment is a long-term observational study monitoring N2O and active microbial communities under regional management practices. And this is an observational study, but this sort of observational study hasn't been done before because we haven't had the ability to track real-time changes. So this would definitely be a first. So we applied ammonia-based fertilizer in order to try to mimic application of what's usually applied in the region, which is anhydrous ammonia. I will be monitoring flux and biological sources in real time for 8 time points per day. I will also be tracking active functional gene groups, substrate sources, and changes in the microenvironment. And also all of looking at functional gene groups on the scale has also not been done yet. And what I will get at the end of this is a figure like this, where on the x-axis we have the day, time, y-axis we have N2O flux. The green dots here represent precipitation, the blue raindrops, wow, represent precipitation events. And each one of these bars is a daily average across all the chambers, with the orange N2O coming from denitrification, the purple coming from nitrification. So you can see that a relationship starts being observed with precipitation events and contribution of microbial communities. Okay. But ultimately, I want to be able to quantify nitrogen loss from the processes across a longer time frame, as you can see here. So here you can see during wetter months, denitrification tends to have play a higher role, which will make sense since denitrification prefers more anaerobic conditions. Then you could see as summer months come, and here summer months are really dry, you can see denitrification contribution slowly decreasing, but also flux decreasing. This could be due to substrate availability or due to the fact that it has been found that denitrification produces overall more N2O than nitrification. And then you can see in September when it started getting wet again, denitrification starts increasing in contribution. Okay, and those preliminary findings are supporting the hypotheses that we found, with denitrification playing a larger role on a 6-month basis, but obviously we're going to be looking at that on a longer time scale. Okay, so experiments 2 and 3 will measure N2O and N2 loss during hot moments of activity for denitrification. There will be lab and field experiments to tease out the mechanisms behind gas production and how real environmental factors are affecting them. So here we have to apply a labeled form of nitrate as potassium nitrate and water to induce environmental and substrate conditions that are favorable. And we need to use this label again in order to track N2 because of the high background. If we weren't using a label, our 15 and percentage would be about 0.3663%. The atmosphere is the same since it's at natural abundance, but the atmosphere is composed of 80%. So regardless of what flux we see with this atom percentage, we won't really be able to pick it up. It will be drowned out by the atmosphere. On the other hand, if we use a label, so this is less than 1%, so labels will look very different. So if we add a label between 10 and 20%, even a small flux could start showing differences, and you should be able to pick up N2 from the soil. And following these hot moments, there will be intensive gas and soil sampling that occur during the first 24 hours. Following that, as activity slows, longer apart time points will be selected. Okay, so then last May, we actually did apply potassium nitrate fertilizer with an enrichment of 10% to these plots. The application is shown in the red line here. The x-axis again, date is time, y-axis is N2O flux. The black line here is total N2O flux, while the dashed orange is N2O flux derived from fertilizer. So this is important to know. It's not all the denitrification that's occurring. It's only the denitrification that's occurring from fertilizer. The rest of these gaps here could be a combination of denitrification and nitrification from natural sources. So we found a couple of things. We found that precipitation events, again, increase activity, as well as increase how much N2O is lost, especially how much is lost from fertilizer. We also found that we can't use enriched substrates to use site preference. The values do not fall within expected ranges. But, and this hasn't been done before, and if we do see trends, which we are a little bit, we might be able to come up with a method to track microbial activity based on which one of the positions of the nitrogen is getting more or less enriched, which can suggest where the substrate is coming from and can suggest which process is occurring. Okay, So to bring everything back together, we all know global population is increasing and food production is increasing. But not only that, the globe is becoming more affluent, and that means we are switching our dietary choices. We're all switching to more meat, and more meat requires more land to feed the animals we're eating. So overall, in the future, we will have to have more arable land. We might try to meet these excess demands by adding more fertilizer. Obviously, there's a cap to how much we can use there. And then we might have to start working on changing our crops. And we know what these main processes are. We know that these processes are going to keep increasing. But again, we lack knowledge of who is actually active in producing N2O. Without this knowledge, we will continue to fail to implement practices, and that positive feedback loop will keep going unless we take steps. And I'm hoping that with this work, I'm going to slowly start identifying what those actual mechanisms are and how they're being controlled by the environment. And with that, I'll take any questions.
Nice job, Milica. That was great.
Thank you.
Yeah, very nice job. Very well done.
Every time I hear you give a talk